So this is the second video for our DNA chapter on nucleic acids and protein synthesis. And in the last video, we talked about DNA, what makes up DNA, and how to build a DNA molecule. So now that we know about a DNA molecule, we can start talking about the processes that involve these DNA molecules. And the first process is DNA replication. So when a cell divides, each new cell must receive a copy of DNA. And the reason for this is because every single one of your cells has a job to do. Every single one of your cells needs those instructions so it knows what to do on a daily basis. I gave the example when we were going over vocab that I can't just give one person the lab instructions and expect the lab to get done if that person's not there, right? Everyone needs a copy of the directions so that everyone knows what to do in case, that one person who had the directions is not available. So every cell needs a copy of the DNA, and your body is constantly making new cells, and each one of those new cells needs DNA, and the process of giving those cells or making DNA for those cells is called DNA replication. This process will occur right before cell division, which we'll talk about next chapter. So here's what happens. Here in red, you see a complete DNA molecule. Obviously, it's short, but it's going to give us what we need here. So what's going to happen is an enzyme with the help of ATP, it's going to come through and it's going to break these hydrogen bonds between the bases. The enzyme that does this is called DNA helicase. And remember, enzymes end in ase, and they're typically named after what they do. So DNA helicase breaks apart the DNA helix. So helicase, that first part means helix. So what's going to happen is when that molecule is pulled apart, you'll see the one part here in red is moved over to the left and the other part in red is moved to the right. And what's going to happen is complementary bases or new base pairs, new nucleotides are going to come in and re-base pair with the existing DNA molecule. The enzyme that helps this happen is called DNA polymerase. So in polymerase, you see the word polymer. So we are building a polymer, which is a larger molecule. So during the process of replication, we are making a large DNA molecule. So this is our end result. You'll see here that the molecule to the left is 100% identical to the molecule on the right. Obviously, yes, we have red-green, and this one is green-red, but that doesn't matter. I have the colors here to show you that this is an original strand, this red, and this red over here to the right is original, and the green sections here are the new nucleotides that came in. But if you look at the actual base pairs, you'll see that they are identical. Here we have AT, over here we have AT. Here we have AT again, here we have AT. GC, GC, CG, CG, TA, TA. So they are identical, and that's what matters. This process will actually happen at many different parts of the DNA molecule because DNA molecules are a lot longer than the one we've shown here. So this is going to happen at hundreds, if not thousands of places within a DNA molecule so that the process could happen pretty fast. Where this happens is called a replication fork. So you have your DNA molecule. When replication is ready, it picks a spot within the DNA and it splits into two. And replication will happen with this strand of DNA, and it will also happen with this strand of DNA, creating two identical DNA strands. So now that we're done talking about DNA replication, we can now move on and talk about another process that includes DNA, and that process is called RNA transcription. So we're shifting gears now, and we are looking a little bit about RNA for a little bit. So remember, the idea of this chapter is that DNA makes RNA, and then RNA will make protein. Now, the reason DNA needs to make RNA is because DNA is trapped inside the nucleus. DNA is not allowed to leave. It will not leave the nucleus. Proteins are made on the ribosomes. So somehow we need to get the material, the message, from the nucleus to the ribosome without DNA leaving the nucleus. That's where RNA comes in handy. DNA will make RNA so that RNA could leave, go into the cytoplasm, travel to the ribosome where that protein could actually be made. And that protein is what is actually going to give the trait. So DNA codes for the trait, RNA sends the message, and then the protein is the actual trait that you're going to see. So let's talk a little bit about what RNA is and what it does. So RNA is a nucleic acid, just like DNA is, but it's a little different in how it's structured. So the first thing that's different here is that RNA is a single strand, whereas DNA is a double strand, so it's only one. Each nucleotide, so it's still made up of nucleotides, and each nucleotide consists of a phosphate, so we still have that. We still have a sugar, except now our sugar is no longer deoxyribose. Our sugar is now called ribose. And we're still going to have a nitrogen-containing base. There are still 4 options, and those options are adenine and guanine, like we had before, and cytosine and, that's not thymine. We have uracil now instead of thymine. So you will not see any T's in an RNA molecule. You will see U's instead. So we still have that circle with a P for a phosphate, the square with an S for a sugar, and you still have your same shapes for bases, but you'll see here at the bottom we have a U in our shape now instead of a T. So everything is still paired correctly. G will still go with C, but now A will fit into U. So we're going to get into this process of RNA transcription. Once again, RNA is formed from DNA in the nucleus. This process happens in the nucleus because DNA is located in the nucleus and DNA is not able to leave the nucleus. So because we need DNA to make RNA, this process must occur inside of the nucleus. We're going to use RNA polymerase as our enzyme and some ATP to help this happen. RNA, because it's only a single strand, that's why it's able to leave the nucleus and travel into the cytoplasm to the ribosome in order to make that protein. Remember, once again, DNA cannot leave the nucleus. Okay, so let's look at what is actually happening. So here to the left, you see in red a DNA molecule. Some things that help me recognize that this is a DNA molecule besides the label telling me it's a DNA molecule. First thing, this molecule has 2 strands. It has a left side and it has a right side. So that tells me DNA. The other thing that tells me this is a DNA molecule is the fact that there are T's within it. Once again, in RNA, we have uracil, so you're not going to see T's. So you'll see here that an enzyme with the help of ATP is going to cut in between the sugar and the phosphate, and it's going to just break the bonds here between these bases, and it's going to sneak out here. So we actually don't need the top and we don't need the bottom for this. And we're going to remove that part of the DNA, and we're not going to use this over here. We're going to use this section here that's now empty. And what's going to happen is new RNA nucleotides are going to come in and bond with this side of the DNA molecule. So you'll see here C is pairing with G. T will still give us an A. So A's are allowed in an RNA molecule. This T is DNA. So we can still have an A. And then this A, you would want to put a T, but there are no T's allowed in RNA. So here we have a U. And this is the portion we pulled out of the DNA, and it's just chilling there for a couple minutes. So now we have a RNA molecule that we are going to remove, and the other part of the DNA molecule that we had previously taken out, we can now put in and put those hydrogen bonds back. So we have now, after this process of transcription, we have our DNA molecule back to normal, And now we have an RNA molecule with our code that is able to leave the nucleus, travel into the cytoplasm, and go to the ribosome to make that protein. So once again, transcription, the purpose of it is to get that code from DNA into RNA so we can take that message and move it from the nucleus to the ribosome to make the protein. So now that we went over transcription, you are ready for worksheet 3. So for worksheet 3, you are going to need a folded piece of paper. There should be some on my front desk, or you could use some of your own. All right, so this is worksheet 3. You are going to take your DNA molecule and you're going to transcribe it into an RNA molecule. So here we have an enzyme that's going to come in and it's going to split those bonds, hydrogen bonds. So we don't even need the top part because the enzyme splits between them, and we don't need the bottom part because I just crossed those out because we're not going to need them. And this section is removed, but we can't do that like we did on the PowerPoint. So I just cross it out in order to help me. So I take a piece of paper, that extra piece of paper, and you fold it in half lengthwise, which we call hot dog. And with that fold, we're going to cover up that part of the DNA molecule we're not using. Okay. So on that, with the fold, I'm going to start to draw in my new RNA bases. So A pairs with T. A pairs with, I typically would want to put a T, but there are no T's in RNA, so I put a U. C pairs with G. C pairs with G again. G is going to give me a C. G with a C. I'm going to just do the other ones. Okay, A with U, T with A, and C with G. So you'll see that molecule. And you'll notice that I did it from bottom to top. For RNA, we do end up doing it from bottom to top. So now, because I have the bases, we still have nucleotides. So each of these bases need a sugar and a phosphate. Sometimes you could use your phosphate going up like we have been doing, or you could put it down. It should go down, but it really doesn't matter which way you make it. So I'm going to fill in all these sugars and all these phosphates. The point is that you have an equal number of sugars and phosphates, just like we did for DNA. So now I make my sugar to phosphate bonds to make sure that this is a chain of nucleotides. And there you have it. That is the RNA molecule. That is transcription. Okay, that will now be able to travel to the ribosome in order to make our protein. You'll notice the space at the top. We won't need that right now, but we will need that when we get to the next step, step 3 of this. So just leave it there for now. But once you have something that looks like this, that is the end of worksheet 3.